students, welcome to Introduction to Nanoscience and Nanotechnology lecture number 24. In this lectures, we will continue our discussions on nanomaterial characterization techniques. We will deal with the imaging methods and imaging methods. We will have a discussion first on transmission electron microscopy, that is in short called TEM, or simply we say TEM. So let's proceed towards today's lecture. Before going to TM, first of all, we should know about the imaging method, that what sort of the imaging methods we have. The first the first category of the imaging techniques that we have is called electron microscopy. And electron microscopy, we basically have two techniques. The first is called TM, that is transmission electron microscopy, and the second is called SEM, or scanning electron microscopy. The second category is called scanning probe microscopy. In scanning probe microscopy, we have three techniques. The first is called STM, that is scanning tunneling microscopy. Second, AFM, that is atomic force microscopy. The third one is called profilometry. So these are the three technique in scanning probe microscopy as a part of the imaging method. So let's proceed towards the electron microscopy. In electron microscopy, first of all, we should discuss about wavelength of the electron. So the wavelength of the electron, just like you can see it here on these particular equations, it depends upon a certain factors. So what are these factors? So these factor in these factor, first of all, we have M. M is basically stand for the electron mass. That is, the wavelength of the electron depend upon the electron mass. It depends upon the electron charge. Uh, then it depends upon the potential difference to accelerate the electron. That is, we have it V. So in more formal words, what is mean by V, or what part we need V? So V is basically potential difference in a way to express the momentum of the electron. I mean, it's some sort of the push. I mean, that is some sort of the energy you want to give to the electron to accelerate it. And this equation is related to the de Broglie equations for the wavelength of the object. Electron can achieve much smaller wavelength than the visible light. So as a result, we can see smaller objects that are not possible to visualize with the help of a naked eye or with the help of optical microscope. So now let's proceed toward the transmission electron microscopy. A photograph of the transmission of the lattice transmission electron microscopy, you can see it here in this particular figure. This is one of the lattice model of the transmission electron microscopy. So what is transmission electron microscope? Transmission electron microscope is a microscopy technique in which a beam of electron is transmitted through an ultra thin specimen, interacting with the specimen as it passes through. It's some sort of the formal definitions. So in this formal definition, we say that transmission electron microscopy is a sort of the electronic device, and this device we have an ultra thin specimen or sample, and that sample we pass by electron beam. So the electron beam, while passing through the specimen, it interact uh, with the sample, and it collect the information, and that information then they are being plotted in the form of a micrograph. So as a result of that interactions of the electron and the specimen, we get an image, and that image has been the result of the interaction of the electron transmitted through the specimen. So after that, when we get the image, so this image is magnified and focused on an imaging device. I mean, when we pass the electron through the specimen or through the sample, so then we get an image, and that image or micrograph is then magnified with the help of the component of this device, that is we call TEM, that we will discuss in the coming lecture. uh, the image which has been formed is first magnified and focused onto an imaging device. Here you can see we have a computers. So that is being plotted here by utilizing some specific software. And along with that, we can get that on a fluorescent screen or on a layer of photographic film or to be detected by a sensor. I mean, it's a possible means for plotting the photograph of the specimen that we want to scan with the help of the electron transmissions. So different component of TEM, you can see it here in this chart. I mean, this is a typical chart of a typical transmission electron microscope. So here you can see that these are the different parts of the transmission electron microscope. So here we will discuss each part or each section in detail. So the first part that we call the electron gun or electron source, you can see it here. It's the topmost part of the TEM. So what is the purpose of the electron source? So electron source or electron gun produces high energy large current, which is highly current electron beam, which is necessary for generating diffraction patterns and high spatial resolution image. I mean, here we have the electron source, and this electron source is normally of tungsten. And the purpose for generating this electron beam is to produce a high energy large current that is current enough for generating the diffraction pattern and high spatial resolution image. Then the second part is being called condenser lenses. So the condenser lenses, what is the purpose of the condenser lenses? The purpose of condenser lenses is to control spot sizes and illumination area on a sample. I mean, these are the function of the condenser lenses. Here you can see all these are the condenser lenses. This is basically called illumination system. I mean, this part from here to here, this is called these are the condenser lenses, and this part as a whole is called illumination system, and their role is to control spot size and illumination area on a sample. Then we have objective lens. What is the purpose of objective lens? So we remember objective lens, I mean, it's the strongest lens in the system, and this lens basically it image the sample. I mean, we put a sample inside the system. So the purpose of the uh, what mean the objective lens, the purpose of the objective lens is basically to image the system, image the sample. And this is the most strongest, I mean, you can call the strongest lens in the system or in the TEM. Then we have, after that we have the intermediate lens and the projector lenses. The intermediate lens, here this is the intermediate lens, and here we have the projector lenses. So their purpose is basically changing modes from diffraction to imaging. I mean, there are two type of modes where we can operate the TEM, that is diffraction mode and imaging mode. So if you want to change the mode of the TEM, so for that purpose normally we utilize intermediate lens and projector lenses. So projector lenses and intermediate lens, they are basically used for changing mode of the transmission electron microscope. And then we have detectors. What is the purpose of the detectors? The purpose of detector is we have various different configurations designed to collect secondary signals produced by the high energy electrons beam. I mean, this is the purpose of the detector. They're normally there in the TEM. These are some of the main component of a transmission electron microscope. So now how it work, how the transmission electron microscope work, I mean, how we characterize the sample on TM. So for that we have to go toward the principle, the working principle of a transmission electron microscope. So we learned about the different part of transmission electron microscope. So now we will learn how these different part they work in a transmission electron microscope. So first proceed towards I mean about the electron gun. So what actually we have an electron gun, just like I mentioned on the previous slide. in electron gun we have a heating tungsten filament. So what is the role of heating tungsten filament? We actually heat this tungsten filament with the help of an applied voltage. So as a result, it generates, electron, and these electron they are being focused on a specimen by the condenser lenses. First of all, we have a tungsten filament here. This tungsten filament is heated with the help of the applied voltage. So as a result of that, it produces an electron beam, and this electron beam one is being generated, then they are being focused on the specimen with the help of the condenser lenses. um, for focusing of the electrons, we utilize normally the magnetic lenses. And why we utilize or why we use condenser lenses or magnetic lenses? Because electrons normally they do not pass through a glass lens. Since the electron do not pass, or electron cannot pass through the glass lens, so therefore we use magnetic lenses for this particular purpose. And the column where we want to accelerate the electron, it should be extra clean, or we say that we should have high vacuum here. There shouldn't be any air particle inside the column. Why? What was the reason behind that? The reason behind that is if we have air particles or any other dust particle, so these particles they will collide with the electron, and we won't be able to get the required result. So in order for the electron to have fully interacting with the specimen, so it's important that we have a high vacuum inside this column, that is called TEM column. So for TEM column there should be high vacuum in order for the electron to fully interact with the sample. So this what happened then? The specimen scatter. We have the electron interact with the specimen or with the sample. So the specimen then scatter the electrons while passing through it, and the beam is focused by magnetic lenses. We have the magnetic lenses. So these magnetic lenses basically focused the electron to form an enlarged visible image of the specimen on a fluorescent screen. So the magnetic lenses, they normally, the purpose of the magnetic lenses, just like we mentioned on the previous slide, is to focus the electron beam to form an enlarged visible image of the specimen on the fluorescent screen. A denser region and the specimen scatter more electrons and therefore appear darker in the image. That you should know about. And in contrast, the electron transparent regions are brighter. So the screen can also be moved aside and the image captured on the photographic film as a permanent record. It's also a function of the uh, transmission electron microscope that we can get a permanent image for our permanent record on a photographic film. That was mainly done on the old system or old model of the TM. That was one of the main feature that we can get image on a photographic film for the permanent record, and even we can do, or we can have the same functions with the latest device, but this is normally not preferred. The most common for today's TM is we get the micrograph, and their micrograph they are being saved inside the computer hard disk with the help of some specific software. So this the photographic film concept that was mainly linked with the some of the previous or old model TM, but it's still valid here in the latest model as well. A typical image that's been taken with the help of TM is look like this. It's a micrograph, a TM image of inorganic materials that is being taken here. So here you can utilize that. Here you can visualize that how a TM image or TM micrograph look like. So this is the TM micrograph of inorganic materials. This how we pictures that how we get the visualization of a material inside the TM. Advantages of TM include that it offers very powerful magnifications and resolutions, allowing that TM have a wide range of applications and can be utilized in a variety of different scientific, educational, and industrial fields for a wide range of applications. TM provide informations on element and compound structures. It's one of the key feature. It not can only locate the sample part, but it can also identify with some of the specific applications that is being linked with the TM. It can also give you informations on the component, on the elemental and compound structures of a materials. Along with that, you can get images on high qualified and details information it can provide in the image. You can have information on the defect structure, that how your sample look like, or whether your sample is defect free or it can have the defect. So all that you can visualize in a TEM micrograph or TEM image. Along with that, DM have some of the disadvantages. These disadvantages are not like something where which we can where which we should avoid the TM. I mean, these are more likely related with the accountability or with hiring of the TM. For example, the first disadvantage we have for TM is that it's very large and very expensive. So you need a specific area, a large lab, and that lab should be only specified for the TM because it requires cooling process, it requires a high vacuum setup, it requires a high voltage source, and along with that, the price of the TM is such that it's not possible for an ordinary lab to buy the TEM. So TEM is very expensive. Along with that, it's large. So this is the first drawback that you can count for the TEM, or the disadvantage of TEM. Then laboratory sample preparations. It's also, you can take that as a headache for the TM. You have to do the tedious job for first preparations. The sample preparation is also a tedious job. You have to do a lot of hard work for your sample preparation before to analyze that into TM. Then operation and analysis require special training. Every person working in the lab cannot operate TM. It requires a person with the specific capabilities, with the specific knowledge and training of the TEM. Only then a person can operate and visualize object if she or he has sufficient training about that. A normal man working, a common man, a layman working in the lab cannot just sit and operate the TEM. So specific training is required, and this also count as one of the disadvantage of the TEM. Sample are limited to those that are electron transparent. We can only visualize those sample with the TM which are electron transparent. If a sample is not electron transparent, so until unless we do not do some track with the sample, we cannot visualize that with the TEM. So that is another drawback of TEM. The last but not the least is that images are black and white. That is, we cannot get uh, the color image with the TM. So whatever you get is basically in black and white because we are taking the electrons. So the electron is basically forming the image for us. So it is black and white. So that you can also count that as disadvantage of the TM. Just like I mentioned earlier, that these advantages, though we have listed, but these are not something by mean of which we should avoid TEM. This is some of the we can say that it is some of the restrictions that a common lab have. We can say that these are the reason by mean of which a common lab, a common material lab, cannot accommodate or cannot get a TEM. Some of the restriction, though we have counted as disadvantages of TM, but these are not sort of disadvantage of TM. One can say that these are the restriction by mean of which an ordinary lab, we can say that the reason that an ordinary lab do not have TM or they cannot require TM. So that's all you have for the TEM. Hope you learned a lot. So stay tuned for the next lecture, because in the next lectures we will have a discussion on the scanning electron microscopy as part of the imaging methods. So stay tuned for the next lecture. That will be lecture number 25. Till then, bye bye.